Namaste. Today we will have a look at two other aspects, cryopreservation and seed banks. Now seed banks are very important for the purpose of conservation, because if you think about a large size tree and you have a limited area, so you can only have n number of trees in your area. But if you could collect the seeds of these trees and if you could preserve them for posterity, so we should be able to regenerate these trees using these seeds. So seed banks become very important as a measure of ex situ conservation. And before we move forward, let us have a look at this paper. It says regeneration of whole fertile plants from 30,000-year-old fruit tissue buried in Siberian permafrost. So what happened in this case is that Siberia is a location in Russia. It is on the northern and eastern portions of Russia, and this area is very cold. So this area has some regions that are a permafrost. Now permafrost is a region that always remains under sub-freezing conditions. So the temperature here is always less than 0 degrees Celsius. So the water is always frozen. So it is permanently frozen, permafrost. Now in these areas, when some excavations were done, then some fruits were found, and when radiocarbon dating of these fruits was done, then it turned out that these fruits were at least 30,000 years old. The exact age was 31,800 plus minus 300 years. So in this case, when the scientists had access to this fruit tissue, they tried to regenerate these plants, and when that was done, the plants were again formed. So these are the plants, they are also giving out flowers, and if we look at the flowers from the related species that is present today, we will observe quite a number of variations from this old sample. So what is happening in this case is that we are having an access to a form of this plant that was existing around 30,000 years back and does not exist today. So this is a very good example of what preservation can do for us. So this is a form of biodiversity that is lost from the earth because it was present quite a long back, but because it was preserved in the form of this tissue, so we can access this biodiversity even after a huge span of 30,000 years. Now this was when these tissue were kept in a very cold condition just by nature. Now this is another example, germination, genetics and growth of an ancient date seed. Now date palms are found in a number of areas, especially in the drier areas, and these are 2000-year-old date palm seeds that were found in Israel, and then these seeds were germinated back. So this is one of the plants that came up through this germination, and then these plants are now growing and they can be propagated. So here also, the place where these seeds were found, these areas were having a number of date palm trees in antiquity, but close to around 800 years back we started getting historical records that there is no date palm found in these areas anymore. So at least for the last 800 years we did not have any of these varieties that were growing here in the antiquity, and the prime reason was climate change. But these date palm seeds were stored in a jar and around 2000 years back, and then because the whole area is very dry and the whole area is these seeds were kept in an area where they were kept cool. So even after 2000 years people have been able to germinate these seeds and get the plants back. So this tells us the power of preserving of the seeds. So in this lecture we are going to have a look at this preservation of seeds in a greater detail. How can we use preservation of seeds as a measure of conservation of these species? So let us begin with what a seed is. So seed is an embryonic plant enclosed in an outer protective coating. So if we look at trees in the forest, so here we have a fruit. When this fruit opens up, it reveals these seeds and then these seeds fall off. So these are seeds. If we cut open a seed, then inside we will find something like this. So we will have a seed coat in the outer area. In most of the cases this is a protective layer, so it will be very thick and very strong. Then we will have some region that has endosperm. Now endosperm is the food source for the embryonic plant. So when this seed falls onto the ground, it will not be able to perform photosynthesis right away. So there is some amount of stored food that is kept for the embryonic plant, and then this is the embryonic plant. These are cotyledons or the embryonic leaves. This is hypocotyle or the embryonic stem. So when this seed falls into the ground and when it gets right conditions, right amount of moisture, right temperature, then this plant will grow out of the seed, and while doing so it will utilize this endosperm for the initial periods till it is able to throw a leaf out, and once this leaf has come out, then it will be able to perform photosynthesis and gain energy from the sun from that point onwards. Now the important point to note here is that because seeds have this stored food in the form of endosperm, so they also act as food for a number of organisms including us. So when we are eating rice, when we are eating wheat, what we are eating is the seed of the rice or the wheat plant. Now similarly, out there in nature there are a number of organisms that eat these seeds, and some organisms even collect these seeds for posterity, such as ants or squirrels. Now if we want to conserve these seeds, we will have to protect them from these animals, and we will also have to protect them from the microorganisms, because microorganisms also want to eat all of this. So how do we do that? So we begin by collection of good seeds. So what do we mean by good seeds? The good seeds have these characters. They are well ripened and healthy. So you should not collect a seed that is half ripened, because in that case the embryonic plant inside might not be well formed and it might not be able to germinate later on, which is our prime purpose when we are creating a seed bank. Then they should be pure and free from inert materials and weed seeds, because finally later on we are going to plant these seeds back into the environment, so we do not want any weeds to come to with it. Then it also should be free of inert materials. So inert materials include things such as soil or things like husk. Now why should we have seeds free from inert materials? Because these inert materials might also be harboring some microorganisms. Then these seeds should be viable and have good germination capacity. Viable means that these seeds should be living. So for instance, if you take a seed and if you say bake the seed, so because of the high temperature the embryonic plant inside would die, and when that plant dies, then we will not be able to germinate this seed any longer. So when we are collecting good seeds for our seed banking purpose, then they should be viable and they should have a good germination capacity. Now it should be uniform in its structure and appearance. Why uniform? Because in a number of seeds that are infested or infected will observe some amount of structural abnormalities. So when our seeds are uniform, then it gives us an indication that these seeds are healthy. Then they should be free from damage and should not be broken and infected by pests and diseases. So free from damage means that when we are processing these seeds, so in a number of situations we will find that we have collected some fruits and then these fruits have seeds inside. So these seeds have to be taken out. So we will have to remove all the pulp that is inside, and then when we have this seed out, then we will dry it and maybe put it into some machine for some grading purposes. Now when we are doing such activities, there should not be any mechanical damage to the seeds, because if there is a mechanical damage, and remember that the seed coat is a protective covering, so if there is a damage to this protective covering, then the seed is very much susceptible to further infections or infestations. So it should not be broken and it should not be infected with pests and diseases. So when we are collecting the seeds, what are the best days for a seed collection? How do we know that our seed is ripe enough to be collected? So there are laboratory methods and then there are field methods. Now laboratory methods include things like the maximum dry weight. So for instance, if you look at a fruit, so initially we will have a flower. Now when this flower is converting into a fruit, so there will be a small sized fruit, then this fruit will grow bigger, and then finally this fruit will attain the maximum size. Now we would find some small seeds here as well, but only in the larger size fruits will we have the seeds that have reached the maturity. So when we look at the maximum dry weight as a laboratory method, what we are doing is that we take a sample of the fruits. So basically we begin by saying day and we have the weight. So in this graph, at day 0 we will have a fruit with a very small weight, and then slowly and steadily the weight will increase, and then later on this weight may remain constant and then it will start falling. Now this is the weight of fruit, because initially we had a very small fruit, then at this point we have the mature fruit, and then at this point this fruit is drying off, or it has split, or it has become infested. So there are some other organisms, say ants or monkeys, that have started eating away the fruit. So in the laboratory method, when we say that our fruit should have reached the maximum dry weight, we want our fruit to be in this range. So this is the maximum dry weight, and when we are using this laboratory method, we want to ensure that our fruits have reached this weight. Next is the chemical analysis of fat and nitrogen content. Now this is important because in a seed that is well developed, we will be having an endosperm that is well developed, so it will be having quite a heavy amount of fat and even nitrogen inside to act as initial starting points for the embryonic plant. So we can perform a chemical analysis of the amount of fat and nitrogen that is there in the seeds. Next, we could go for examination of embryo development and endosperm of sample seeds through X-ray radiographs. Now in this case, what we are doing is that we take a sample of the seeds from the fruits, and then we perform an X-ray examination of those to understand whether these parts have fully developed or not. If they have fully developed, that means that our fruits are now ready to be collected for seeds. Next, you have moisture content of fruits. So in a number of cases, the moisture content will also tell us whether these fruits have ripened fully or not. Then you have the field methods. So field methods include density of fruits or also weight of fruits, color of fruits. So for instance, in a number of fruits, you will observe a change in color from green to say yellow or green to red, and that will tell us whether these fruits have matured fully or not. And also a visual examination of seed contents after cutting. So what we are referring to here is that in a number of seeds such as bamboo seeds, so you will have this husk, but inside there will not be any seed, and in certain situations when this fruit is completely developed, you will have a seed inside. So you can always take a few samples out and observe what is inside the seed. Do you have your embryonic plant inside or not? So that can also be done. Next, which trees should we collect our seeds from? So we want to develop a seed banking facility. So which trees should we collect our seeds from? So the first point is that you should collect from dominant or codominant trees. Now dominant or codominant trees means trees that are the highest that are there in that particular stand. So a dominant tree has come out and is getting sunlight from all the directions. So you should collect seeds from dominant or codominant seeds because they are the largest size trees and they are also considered as having the best genotype. Then you should collect from a minimum of 10 seeds at a time, preferably from 25 to 50 trees. So you are not collecting just from one particular tree because you want to have a diversity of genotypes that are there. You should collect from trees that are far from each other to avoid collecting from half siblings or parents. Now in this case, what we are referring to is that if you have a tree and if you try to understand that if there is a fruit or a seed here, and when it gets dispersed, what is the maximum distance that it can go out? So for instance, if this is the maximum distance to which you can have the seeds or the fruits of this plant or this tree, so the next tree that should be collected should be outside of this distance. So this should be the next tree. Why? Because if you are collecting from a tree that is here, then this particular tree, so let us call this tree 1, tree 2, and this is the third tree. So if this is within this distance, so it is quite possible that T3 is the progeny of T1. So essentially if say this tree was a female tree and it was getting pollens, so these fruits got these flowers got pollinated, they formed the seed, and then this seed fell here and this formed our T3. So in this case, when we are collecting seeds from T1 and T3, the amount of genetic diversity between both of these collections will be less because they are related to each other. Whereas when we are collecting from T1 and T2, then because they are very far from each other, there is a reduced possibility that both of these trees are related to each other. So in that case, the amount of genetic diversity between both of these will be much greater. So you should collect from trees that are far from each other to avoid collecting from half siblings or parents. Now before collecting, mark the individual trees and create a record for these. Collect equal number of cones, fruits or seeds per tree. Now why is this important? Because essentially what we are doing is a sampling procedure. So for instance, if we collected say 1000 seeds from here and say 10 seeds from here, then this will not be representative of what is being found in the nature. In nature we are having one tree that is T1 and one tree that is T2. So if we are collecting 1000 seeds from here, we should also be collecting 1000 seeds from the second plant. So this is what is being said here. Collect equal number of cones, fruits or seeds per tree, whatever you are collecting. Next, mixing of seeds can be done for large scale collections. So it is not necessary that you should keep the seeds of T1 separate from those of T2. You can mix all of these depending on the conditions. Now when we are especially collecting for seed banking purposes, then we generally increase the number of seeds or fruits that needs to be collected. So in place of 25 to 50, we generally say let us have even more number of trees per gene pool, 50 to 100 trees. And in this process, dominant and codominant trees are preferred, but we also might go for a random sampling including poorer than average trees, because even those poorer trees might be having some genes that are important for biodiversity. So it is quite possible that our tall and dominant trees are having less amount of disease resistance, but our shorter trees are having a better disease resistance. So when we are collecting for conservation purposes only, so in that case a random sampling is preferred, including those of poorer than average trees. Next, collection in better seed years for better representation of parents. Now what is this thing? So when we talk about a seed year, so essentially if you have these trees in the forest, now the seeding can happen in two ways. One is a sporadic seeding. So in the sporadic seeding, what happens is that in a particular year this tree might be having fruits and this tree might be having fruits, but the other trees are not having fruits. Or it is possible that a number of these trees are having fruits, but the number of fruits that are being given out are very less. So typically in the forest we observe that in a period of say 3 to 5 years, there is 1 year in which we will have the maximum amount of fruiting and seeding, and then this will remain subsided for another 2 3 more years, and then it will again peak up. So we will observe a graph such as these. So here we have the number of seeds or number of fruits per tree, and here we have the time, or let us call it the year. So here say we have the year 1990, 1995, 2000, 2005, 2010, 2015 and so on. Now what happens is that typically we observe that there is a peak and then we will have a break, then another peak and then another break and then another peak and then another break and so on. So these years in which we are having the highest number of seeds or fruits per tree are known as seed years. So this is one seed year, this is another seed year, this is another seed year, this is another seed year, and all of these seed years show a certain periodicity. So essentially if this period is say 7 years, then this period will also be around 7 years, this period again will be around 7 years. So when we are collecting in better seed years, what is happening is that we are having more number of of fruits per tree and also more number of trees that are fruiting. So essentially we have a larger number of seeds. When we have a larger number of seeds, then the amount of diversity is also greater, because in our previous situation here, if we collected in this particular year, we would only have the diversity of these two trees. But when we are collecting from our good seed year, what is happening is that this tree is also fruiting, 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 and this tree is also fruiting. So in that case, we will have the diversity of all the trees together. So which is why a good seed year is preferred for the collection of seeds. Next, larger quantity of seeds to be collected for each provenance. So a provenance means a region. So for instance, in the case of teak, you can have teak from Madhya Pradesh, you can have teak in Kerala, you can have teak in Tamil Nadu, you can have teak in Karnataka. So all of these are different locations from which you can have the teak seeds. And when we are collecting for the purpose of conservation, when we are collecting to create a seed bank, we want to have as many seeds as possible, so that for each sample, for say every species like teak, if we have say around 1000 seeds, then it is normally observed that the viability of the seeds goes down with time. So this is viability and this is the time. So right after collection you will have the maximum viability, but then later on it will go on decreasing and finally it will reach a viability of 0 after say in this case let us say this is 100 years. So this is the viability curve. Now if you are collecting a very less number of seeds, then with this graph you might have a 0 viability at the age of say 30 years. So we always want to have a larger number of seeds per provenance, so that even after a long period, even after a very long period has elapsed, we still have some seeds that are viable and that we can use to propagate the species. Now when we are doing the seed collection, you need to organize a number of things. We need to organize collection teams. So these collecting teams have to be taught about how this collection needs to be done, what are the do's and don'ts that should be done. So for instance, if there is a tree and if there is a seed that has fallen down into the ground, so after it has fallen into the ground, it might be covered with dirt, it might be having some fungi together with this dirt. So this seed should not be collected, whereas seeds that are still there on the trees are better. So things such as these have to be taught to all of these collecting teams. Then organization of transport, because you do not want to keep seeds out there in the field, we need to bring them to our facility as soon as possible, so that they are preserved in a better manner. So organization of transport is required. You might require some equipment, so equipment for climbing of trees, equipment for plucking of branches, equipment for depulping of seeds, all these equipments are may be required. Organization of records, so for every sample you need to know what is the species, where this species was collected, when was this particular sample collected, and so on. Organization of permits, so this is especially important when you are moving your seeds across the international boundaries. So for instance, if you want to send your seeds to say Norway, so there might be a number of permits that are required, especially from the biodiversity point of view. And then organization of seed extraction from the fruits. So once you have brought your seeds or fruits to the facility, next you need to extract the seeds and process them. Now what are the ways of collecting the seeds? So you can collect seeds after they have fallen out naturally, so which is known as the natural seed fall. The second is manual shaking. So you can go to a tree and you can shake that tree so that all the fruits come down, and then you can collect them. Or you can make use of an equipment for mechanical shaking of trees, or you can make use of tree funnels. Now tree funnel is an interesting concept. So here what we do is that if there is this tree that is now giving out the seeds, what we do is that we construct a funnel around this tree, and this funnel is normally made out of cloth. So these are say some poles. So this is a pole and this is the funnel made of cloth or a plastic sheet. Now what would happen in this case is that if there is any fruit on the tree, let us represent it by a red color. So these are the fruits, and when these fruits break open or when these fruits fall down, so they will fall on this funnel and they will get collected here. Now this is especially important for those fruits or seeds that are small in size, because in the case of these small sized fruits or seeds, your fruits and seeds might disperse to a very large distance, and then once they have mixed up with the dirt that or the soil that is surrounding these trees, then it becomes very difficult to collect them back. But then with this mechanism, your seeds will fall into the funnel, they are not getting in contact with the soil, and so they are not getting into contact with the microorganisms. So once your seeds have collected here, you can take them away. So this is specially used for conditions such as cones. So in the case of pine cones, their seeds are very small, so this is one thing that could be used. Another thing is raiding of animal caches like those of squirrels and ants. Now these animals, squirrels and ants, they collect a number of fruits and they collect a number of seeds, and then they bring them to their own homes, so to speak, and then they collect them there or they store them there for weathers that are not very good. Now in these cases, if you raid a squirrel cache, so what you will find is seeds from a number of different trees surrounding those areas. So this could also be one way that can be used. Normally we do not go for these because the places where these animals store the fruits and seeds are generally damp, and in those damp areas there is a very high probability that you could have a fungal infestation. But then talking back about our first example, the Siberian permafrost, there the fruits were collected from one of a squirrel cache. So that is one example where we collected seeds or fruits from a very old sample. Another is collection by plucking. So in the case of plucking, you can climb a tree and then you can pluck different fruits once they have matured, or you can have collection by cutting, breaking and sawing. So in this case, you can cut a branch or you can pull a branch to break it, or you can saw a branch to break it, and once this branch has come down, you can then collect the fruits from this branch. Now once your fruits have been collected, there are a number of other operations that need to be done. One is depulping. So in this case, you are separating the pulp out from the fruit. Now pulp is a soft tissue, it has a lot of moisture, and so there is a larger chance that your seeds might get infested if they are surrounded by the pulp. So if we remove this pulp, this can be done by hand by soaking the fruit in water or by using a mechanical device. Next, after pulping, you could go for a drying under the shade or a sun drying. Depending on the species, depending on the seeds, you will have to dry the seeds. Now why is drying done? Because typically our seeds have a larger moisture content, and at larger moisture content, the probability of the seeds getting infected or infested becomes larger, whereas in the case of dried out seeds, there is hardly any microorganisms that can grow there. Now a case in point here would be the date palm seeds that we saw earlier. So even after 2000 years, they were able to retain their viability because they were stored in a cool and dry place. Now in certain situations, if you need to dry these things very fast, or when the weather is not supporting, we can even go for drying with artificial heat or using kilns. Now in the case of some seeds, you have wings attached to the seeds. So in this case, you will have a seed. So this is a seed, and then there is a wing attached to it. Now why are these wings attached to the seeds? Because remember, you have this tree, and this tree had a fruit, and this fruit had a seed. Now if this fruit breaks open, the seed will directly fall down, whereas if this seed has wings attached to it, so if this seed has these wings, then when there is even a small blow of wind, these seeds will be able to disperse to a farther off place. So just for facilitating dispersion, there could be a number of seeds that have these wings. Now once you have collected these seeds, there is no purpose of having the wings together, and besides, if you have a seed with the wings, so the later processing might become difficult. So for instance, if you want to grade these seeds, so you for instance you wanted the bigger size seeds in place of the smaller size seeds, because the larger size seeds are more mature, they are having more amount of endosperm. Now in that situation, when you are putting your seeds through a grader, then these wings may come and block the holes or block the seeds. So in those situations, we may go for a de-winging operation, so both of these wings are taken off. Then you could have a threshing and sieving operation, so as to remove all the husk or all the broken parts of the wings or the broken parts of the fruits that are together. So you can have this threshing and sieving operation or a blowing operation. Then we can go for a sorting and a grading operation. Now in sorting and grading we do two kinds of things. One is a visual examination, in which you ensure that your seeds are not having any defects, especially they are not broken down mechanically, their seed coats are intact, they are not showing any signs of infestation. So for instance, if you have a seed, if there is a seed and then there is a black colored spot here and a black colored spot here, so this might show that there is some amount of disease that is coming up on the seed. So there is some amount of microorganism growth that is going on in the seeds. So in the grading operations and in the sorting operations we remove these seeds. The second sort of grading operation is when you sort your seeds by size. So in this situation what happens is that you have a sieve holes. So this is a sieve in which you have holes that are there in different gradation. So this one is a small diameter, this one is a larger diameter, and this one is the largest diameter. Now when you put all your seeds here and this particular sieve is shaking in this manner, so now when all of these seeds fall and when they move down, all the smaller seeds will come out through this hole and collect in one bucket. The larger size seeds will come down from this hole and collect in this bucket, then this bucket, and then this bucket. Now these seeds are the largest size seeds and these ones are the very small size seeds, and then these are the medium size seeds. Now because these seeds have the largest amount of endosperm and because these seeds have reached the complete maturity, so the amount of viability in these seeds and also the amount of regeneration that we will observe from these seeds is much greater typically than these smaller seeds. So because of this we can go for sorting and a grading operation. So these are different operations that need to be carried out. Now how long does a seed last? What is the longevity of a seed? So depending on the natural longevity of tree seeds, we divide them into three categories. Microbiotic, in which the seed lifespan does not exceed 3 years. So micro is very small life, medium sized life, and very long life or large size life. So mesobiotic is 3 to 15 years and macrobiotic is 15 to over 100 years, so which has a very long life. Now why do different seeds have different life forms? So we can understand that by looking at this classification. So different seeds can be classified as orthodox seeds or recalcitrant seeds. Now orthodox seeds are seeds which can be dried to a low moisture content of around 5% and successfully stored at low or sub-freezing temperatures for a very long period. For example, grass seeds. Now typically in these seeds the amount of fat content is very less or the amount of oil content is very less. So what happens is that if you have any amount of oil in a seed, so the oil, when it comes in contact with oxygen, then it becomes a rancid oil. Now rancid oil means that this oil has undergone oxidation. In the case of oxidation, a number of bonds have been broken, and after breaking, oxygen atoms have come into the structure. So essentially the purpose of the oil is lost. Now if you have those seeds that have very low amount of oil, you can dry these seeds and then keep them for a very long period. So these are known as orthodox seeds. So even our seeds such as wheat or rice come in this category. So you can store these seeds for a very long time. The second category goes by the name of recalcitrant seeds. Seeds which cannot survive drying below a relatively high moisture content, often in the range of 20 to 50%, and which cannot be successfully stored for long periods, example sal seeds. Now these seeds have a very high amount of oil content, and so when you are drying these seeds completely or you cannot dry them to a high level of dryness, because these typically have quite a heavy dose of oil, and during this process of drying the oil also becomes rancid. So these are recalcitrant seeds and you cannot store them for a long period of time. So this is the natural variation, but then the longevity of seed also depends on how it is being stored. So, even in the case of an orthodox seed such as a grass seed, you can have a situation in which your seed does not have a long longevity. For instance, if you store it in a damp place so that fungus grows on it, or if you have collected these seeds before they have matured, or if during processing these seeds have broken up. So, what are the factors that affect longevity in storage? So, we have the seed condition, the maturity, mechanical damage, fungi and insects, and the initial viability. So, these are the 4 initial conditions. Then the age of seeds. Now, longevity also depends on the age, because typically the viability goes down with the age. So, here you have the viability and here you have the age. Now, the viability will typically go down with age. So, for instance, if you consider your longevity as viability of greater than 5%. So, this is 5% viability. So, if you have these seeds, if you have started at this point, then your seeds will typically last for this much period. Let us say that this is 70 years. But then if your seeds have already aged till this point, and then you are considering the longevity of these seeds, then this age might only be 30 years. So, the longevity in the storage also depends on the age of the seeds. So, the newer these seeds are, the better it is. And then it also depends on the storage conditions, the atmosphere. So, the level of oxygen should be low, not only because oxygen can oxidize any of the oils that are there in the seed, but also because oxygen is required for respiration, and if your seeds are respiring, so they might end up using all the endosperm. The amount of moisture should also be low, because with moisture and good amount of oxygen, you can have more amount of respiration, and also if there are any pests or any microorganisms, they will also be able to survive well. Temperature should be low. So, for instance, in both our earlier cases, in the case of the Siberian permafrost, the seeds, the fruit was always kept below 0 degrees Celsius. So, it was always kept frozen. So, when something is frozen, then there cannot be any life going on. And even in the case of the date palms, they were stored in jars which are kept in a cool position. And then there should not be any light here, because again in the case of light, you can have some seeds that might start germinating, or you might support some other life forms. So, light should also be absent. So, in most of the situations when we are creating a seed bank, what we do is that we collect the best of the seeds, we collect them when we do not keep them for a very long period of time, and then we seal them hermetically in a glass container, say an ampoule. So, what would happen in that case is that you would have glass ampoule. In this ampoule you would put all the seeds inside. So, now you have all these seeds, and once your ampoule is full, you will then remove the air from inside. So, the air is removed out, and then this portion is hermetically sealed. So, it is now completely fused, and so your seeds do not have any more access to oxygen. And then these ampoules are stored in dark conditions and at low temperatures, and typically they are stored at sub freezing temperatures less than 0 degrees Celsius. So, this is one example of one such storage facility. So, this is the Svalbard Global Seed Vault in Norway, and this location is chosen in a way that we do not have any earthquakes in this region, there is no tectonic activity. This area is on a very highland, so that if there is global warming, if all the ocean levels rise, even then this area will not be inundated. And then this is one area that is always under a permafrost, and even then the inside temperature is cooled even further, and even if all the power supply goes off, then the outside is already sub-zero, and it will take a number of centuries to reach a temperature of 0 degrees Celsius. So, this is one way in which the seeds are stored. Now, the underlying principles of seed banking are these. If you have a seed bank and if you do not know which seed is which, then it does not make any sense. So, identity of accessions has to be very clear. So, we make use of passport data together with the herbarium voucher specimen for identification. Now, what is a voucher specimen? So, in the case of a voucher specimen, you would have a sheet of paper in which you would have a pressed flower. So, this is a pressed flower, a dried flower with a leaf or maybe a fruit, and all of these things are dried and all of these are pressed just like a herbarium sheet. So, when you have this sheet, and then you will also have the identification details. So, the species, the date of collection, the place of collection, collected by whom, and so on. So, all these information are there. So, this becomes herbarium voucher specimen. So, this is always supplied along with the seeds, so that we can have a very clear identification of what this species is. Next, if you have stored your seeds and these seeds die off, then it does not make any sense. So, you need to maintain the viability, and you also need to maintain the propagability. Now, propagability is the property of these seeds being able to propagate the species later on. So, essentially, if you have a seed that is viable, but the plant that it produces is infertile, so it won't be called a high level of propagability. So, the seeds should be in a position to that they are living and they are also able to propagate the species. Next, you need to maintain the genetic integrity. Now, genetic integrity means, as we have seen in our fundamentals lecture, that after a while the amount of genetic variations may go down. So, we need to maintain that. So, how is this maintained? So, for instance, after your seeds have gone down in their viability, so you need to, so this is the viability and this is the age. So, after your seeds have gone down in viability, so here it was 100% and say this was 50%. So, at 50% you want to have more seeds into your facility, because you still do not have a situation of doomsday. So, you can still collect these seeds back. Now, you have got two options. One option is that you take these seeds out, you grow them in the field, produce plants out of them, and then recollect those seeds and then place them back again. The other option is that you collect these seeds right from the natural sources. So, you collect them back from their forest. Now, if you are replanting these seeds, so the amount of genetic variation will go down after every such replanting, because you have this small collection of seeds, the small which will give out a small collection of plants, and you will only have breeding amongst these. So, very soon inbreeding might crop in. But if you are collecting these seeds right from the nature, then the amount of genetic integrity is much greater. So, when you are maintaining a seed bank, you also need to ensure that the genetic integrity is maintained. Next is maintenance of germplasm health. So, you need to ensure that the seeds are free from diseases and pests. Next is physical security of the collection. So, including safety from earthquake, floods, fires, global warming, and even terrorist activities these days. So, you need to have a good amount of security in this area. Now, if you have this germplasm and you are just storing it and you are never going to use it, it does not make any sense. So, there needs to be a process of availability and use of this germplasm. So, if somebody wants to have these seeds, what are the protocols that would have to be followed? These need to be very clearly defined. And also the availability of information. So, availability of information means that if you have a seed bank, then you should also have a procedure through which people might come to know what all seeds are there, what all seeds are required, what are the ages of those seeds, what are the provenances of those seeds, and things like that. So, all of these things are being taken care of in the Svalbard Global Seed Vault. Now, here we talked about the plant cells and the plant seeds, but then similar low temperature principles are also used for animal cells and tissues, such as sperms, ova, embryo, tissue samples, and so on. So, for instance, in the beginning we saw that using the fruit tissue, plants were raised back from 30,000-year-old samples. Now, if you have animal tissue and you can make use of cloning, you can recreate this animal later on if you can store it at a sub-zero temperature, at freezing temperatures. Or if you have stored sperms and ova together, you can fuse them together later on and create an embryo, or you can just store the embryos as such. So, all these things are not just applicable to plant cells or plants, they are also applicable to animals. And so, we have also created a number of cryopreservation facilities for animals. So, this is one such facility. So, these are all large size chest freezers in which we are keeping different samples of animals. So if you open one of these, this is how it will look. So with this lid open, you have these insulating materials, and then you have all these different chests in which a number of samples are kept. So how does the sample look? So this is one sample when you have taken it out. So this is a plastic vial which is airtight, and this has an identification code. So it says 17 by 598. So this is an elephant adult female, and then this was collected in August 2017, and this tissue is from the head region. Now such kinds of facilities are important not just for preserving of these variations over a long period of time, but they also become very crucial when you need to understand any diseases. So for instance, if in your area you find out that there is a particular infestation of, say, tuberculosis, now you want to ask, when did tuberculosis actually come into my park? So in that case, if you have one such facility, you can go back to all of your stored samples, and then you can investigate when was the first instance when you are finding a tuberculosis bacterium in your sample, or if when you are finding antibodies against tuberculosis in the blood samples. So things such as these can also be done. So cryopreservation is extremely important for plants as well as animals, and not just for their propagation, but also for as a repository for biodiversity, and also as a repository for further scientific studies. So that's all for today. Thank you for your attention. Jai Hind.